Hello everyone, a very warm good morning to all of you. I am Akanksha, student of Banas Hindu University, and my presentation topic is Nutrigenomics, a diet for your DNA. The term nutrigenomics came into scenario after the Human Genome Project, which was initiated in 1990 and got completed in 2003. It took almost 13 years. So, in 2001, the Pellegrin was the man who coined the term nutrigenomics, and the interrelationship between food and genome was established in 2007. The primary goal of this project, Human Genome Project, was to discover the complete set of human genes and make them accessible for further biological study and determine the complete sequence of DNA bases in the human genome. Nutrigenomics. Nutrigenomics promises the way your innermost self or your DNA talks to your food. It is a step to move beyond the love-hate relationship with your food. It is more than that. As we all know, food is meant to have positive effect on your body, on the activity of DNA, as well as it is also meant to create a shield for health. But we came across such situations where we came to know that one person's superfood is another person's poison. How? It is because of the way your gene is responding to it. It totally depends upon your genetic makeup. So, broadly defining, nutrigenomics is the relationship between nutrient diet or food and the gene expression using modern tools like omic technology, which includes transcriptomics, metabolomics, epigenomics, and proteomics, and the study of how food affects a person's genes and how a person's gene affects the body, the way the body responds to food. We are dealing here two terms, nutrigenomics and nutrigenetics. In nutrigenomics, we deal with how does what we eat affect our genes. Here we are dealing with the how food is affecting our genes, while in nutrigenetics, how does a person's genes impact on individual response to diet. Nutrients also acts as a dietary signals. Here nutrient factor which can be micronutrients or macronutrients like vitamins or minerals or carbohydrates or proteins, and then transcription factor occurs where the transcription occurs, and then gene transferase which further leads to energy homeostasis, that is the balance of energy inside the human beings, and the nutrient absorption as well as cell proliferation. Common dietary chemicals which acts on human genome either directly or indirectly can alter gene expression, and under certain circumstances it has been found that diet can be a serious risk factor for a number of diseases, and some diet regulated genes are likely to play a role in onset or progression or severity of chronic disease. The degree to which diet influences the balance between healthy and disease states may depend on an individual genetic makeup. Dietary interventions based on knowledge of nutritional requirement, nutritional status and genotype can be used to prevent, mitigate or cure chronic disease. The bioactive components influences the genetics, and here it is described how. Nutrigenomics works on DNA, nutritional epigenomics and nutritional transcriptomics works on RNA. Proteomics is a detailed study of protein, and metabolomics is a detailed study or a large scale study of metabolites or small molecules. In transcriptomics, there is a study of transcriptome, which is a complete set of RNA, also known as expression profiling, which allows for identification of genes and pathways which respond to and also counteract between biotic and abiotic environmental stresses. In proteomics, large-scale study of protein occurs. In epigenomics, a study of epigenetic changes in cells occurs, and the changes are in the way like genes are switched off or on without changing the actual DNA sequence. And in metabolomics, large-scale study of small molecules like metabolites in cell tissues occurs, and these small molecules and their interaction within the biological system is known as metabolome. And this DNA, RNA, protein and metabolome leads to phenotypic characterization. Interrelationship among specific nutrients, genes and health. Green tea, the biological name is, scientific name is Camellia sinensis, And the potent antioxidant catechins and epigallocatechins prevent certain cancers and heart disease. Some women appear to show a reduction in breast cancer. Curcumin, which is a yellow pigment from turmeric, Curcuma longa, a basic ingredient in curries, produce suppressing genes for enzymes that produce inflammatory prostaglandins, and that particular enzyme is cyclooxygenase 2. Prostaglandins linked to colon cancer, heart disease, arthritis, and Alzheimer's disease. Indian peoples are very fond of curry, and as we know that, and curry eating people of India have the world lowest incidence of Alzheimer's disease, as stated by a professor of neurology, University of California. Now we deal with gene diet disease interaction. About 97% of the gene have known to be associated with human disease result in monogenic diseases. Modifying the dietary intake can prevent some monogenic diseases. In phenylketonuria, food containing amino acid, specifically phenylalanine, and including high protein food should be avoided, like fish, cheese, eggs, milks, chickens, dried beans, nuts, and tofu. In the same way, alcohol must be avoided in those individuals where defective aldehyde dehydrogenase enzymes are present. Similarly, patients or individuals having galactosemia, a condition where they lack galactase enzyme, should avoid diets which contain lactose or galactose, and lactose or galactose is majorly found in milk and meat products, so they should try their best to avoid these diets. Now we are dealing with the case study coffee and caffeine, which was conducted by Anderson et al. In 2013. Caffeine metabolism occurs in liver, where it is rapidly absorbed within an hour following ingestion, and its mechanism of action involves increased excitability in the brain by manipulation of a pre-existing lock and key mechanism. When caffeine is consumed, it blocks adenosine, which acts as key here, from binding to its receptors, which acts as lock here. This benefits athletic performance by reducing feelings of fatigue and perceived exertion thresholds, which can improve endurance and leading to performance improvements. Adenosine is a natural occurring chemical in the brain, and that binds to receptor sites to slow down neural activity and increase feeling of tiredness. The advantage and disadvantage of nutrigenomics are included here. The major advantage of nutrigenomics is like improving health and preventing disease through tailored diet or customized diet, or we can say personalized diet, and lifestyle prescriptions. Effectively prevent and manage chronic disease, and helps to reduce healthcare cost, and also a better understanding of mechanism in disease susceptibility. And the disadvantage of nutrigenomics are like focusing on specific nutrients or foods, that means more conscious about your food, increased cost associated with personalized diet and designer foods, and on customization of either your foods or costumes or your home, the cost will increase. Nutrigenomics depends on the recent development of advanced technology that allow us to process a large amount of data relating to gene variants. Most chronic diseases are not caused by monogenic mutations or single genetic effects affected by a single dietary exposure, but by a complex interaction among a very large number of different gene variants. Now, the knowledge of how food interferes with the genetic code and how the organisms respond to these interferences and with the phenotype can be clarified and identified clearly. The ultimate aim of this emerging field of science is prevention rather than cure. This is very similar to the dictum of father of medicine Hippocrates, who said, "Leave your drugs in the chemist's pot if you can heal the patient with food." Thank you so much for your attention. Bon appetit.